my name is Laura Witt, and I am getting my master's degree in science education at the University of Northern Iowa, and today I'm going to be talking about the transport system in plants. Plants use the transport system to move things from one place to another, either from the roots to leaves, or from leaves to the roots, or from leaves to the other leaves. They transport things like minerals, sugar, hormones, and most importantly, water. So how do they transport these things? They use a system of pipes called the xylem and the phloem. The xylem transports minerals to their primary destination and water, while the phloem redistributes minerals, transports sugar, and hormones. So the xylem's most important job is to transport water. Why does it want to transport water? For transpiration. Transpiration is the act of moving water into cells and making those cells swell up and open pores in the leaves called stomates, and this allows gas to be exchanged. So the plant is producing oxygen and it's using up carbon dioxide to make sugar. And so it uses water movement to open up holes in the leaves and cause this gas exchange to occur. But how does water get from the root system, where you water your plants, up to the leaves to open up those pores? It's either pushed or it's pulled. So it's either pushed from the roots up to the leaves, or it's pulled. So today we want to explore exactly which is occurring. So what's the difference between the push system and the pulled system? Well, let's talk about pushed first. There could be either two mechanisms by which water can be pushed from the roots to the leaves to open those important cells so that gas can be exchanged. It could either be done by a pump, so some kind of pumping mechanism, kind of like a heart, could be pumping water from the roots up to the leaves. Or it could be done directly by the root system. Something in the roots could be pushing water, again, up to the leaves. Initially, when a plant first starts developing, it is pushed by the root system. But we're not sure right now if that's also the case for transpiration or not. So what about pulling? If water is being pulled from the root system up to the leaves, what would that look like? We don't really have a system in our body where we're pulling things up like a heart, but it could look something like a rubber band. It could look something like a rubber band. At the bottom of the roots, all the way up to the leaves, water could be stretched and under tension just like a rubber band. So that's what that system would look like. As water was leaving the leaves, more water would be pulled up from the roots and it would be under tension. So how can we test these two mechanisms? What can we do to see whether or not water is being pushed or if water is being pulled from the roots to the leaves? What I think we should do, and I'm kind of in charge of this project since you're just watching a video, is I think that we should take some kind of dyed water, maybe blue. Blue is my favorite color, and you don't really see blue in nature very often, so we would know that this blue dye would be coming from the water. And inject the blue dye directly into the xylem and see what it looks like. See what happens to the dye, whether it moves one direction or another direction, and maybe that'll tell us something about whether water is being pushed or pulled. So if water is being pushed, what exactly would we observe? I drew this picture up here, and here's the xylem in the middle with water being pushed in one direction only. Whether it's being pushed by the root system or by some kind of pumping mechanism, doesn't matter. The outcome's the same. Water is being pushed in one direction. Here's a syringe with the blue dye. What happens when you get an injection from a doctor? directly into your vein? Yeah. So if it goes directly into your vein, your heart pumps it in only one direction. It doesn't go both directions up your arm, it goes one direction through your vein in your arm. Same thing would happen here. As we injected the dye would be pushed along with this water, and we would see the dye traveling in one direction only. So that's what would happen if the system was being pushed. But what if the system was being pulled? In this system, what do you think we would observe? Here's the xylem, and again, here's the syringe injecting blue dye into the xylem. If the water started in the roots, which we're assuming is down here somewhere, and it's traveling up towards the leaves, you would expect it being pulled like this rubber band. So what would happen if I were to cut the rubber band and inject the blue dye? So here's the rubber band representing water under tension. Water being pulled from roots to leaves. And here's the scissors, which is going to interrupt the tension by cutting the rubber band and kind of represents our syringe. Since when we insert this syringe into the xylem, it's going to have the same effect as these scissors because it's going to interrupt the flow of water. So here's the water down in the roots, and when those stomates open up in the leaves, that forces water to be pulled up to the leaves. Now, this is when transpiration is going on. We have water at the roots and water at the leaves, and it's under tension because it's being pulled in this model. If we were to inject our syringe, we would interrupt this pull, like that. Just like the rubber band example, in plants, if we were to insert a needle into the xylem, we could expect the same kind of spring reaction. So as we inserted the needle, the water would break just like the rubber band, and as the dye entered, some dye would go one direction and some dye would go another direction to fill that void. Okay, so let's review. If plants push water from the root system to the leaves by either a pumping mechanism or by pushing water from the roots, then when we insert a dye into the xylem, we would expect to see the dye to go only one direction, towards the leaves. However, if water is being pulled from the roots to the leaves, when we insert a dye, we would expect the water to spring apart just like that rubber band example, and we would expect to see the dye move in two distinct directions. Okay, so we're at my house today and we're going to try the procedure that I outlined, just a minute ago in the video. So let's go over the materials that we have, then I'm going to show you the general procedure, and then I'm actually going to do the procedure. After I do the procedure, I have like a slideshow of high-resolution pictures of the results that I got, and then the video will be over. So, okay, let's go. Here we have all of the materials that we're going to need for this procedure. Starting over here, we have water-dyed blue, and we also have some red food coloring here as well, because while I said blue doesn't occur in nature, so I like to use blue. I'm also going to try using red because it will give a better contrast to the green. Next, we have a open syringe, so no plunger, and a butterfly needle. And when I add blue dyed water to this, it's going to just drip out using gravity. So I'm not going to be pushing in with the plunger at all. Next, I have a clean razor blade. Then I have this plant, which was supplied to me by UNI's greenhouse. And finally, I have the sun, which is necessary for transpiration. So here's the syringe, and now it's filled with blue dyed water. And as you can see, holding it like this, the dye doesn't escape, but when I flip it over, it starts to drip out slowly, and that's because of gravity. And I can control the rate that it moves out by lowering or raising the syringe. So all the way up at the top, it drips the fastest, then as I bring it down, it slows down. So what I'm going to do is I'm going to insert this syringe into one of the stems of the leaves. Then I'm going to remove the syringe, and using a razor blade, I'm going to cut out the stem around the area where the needle penetrated to stop transpiration from occurring and messing up our results. Then, after that, I'm going to take the piece that I excised and I'm going to remove this outer layer so that we can have a better view of the xylem and see what happened to the dye. So let's try the procedure that we outlined. Starting here, let's insert the needle into the stem and then take it back out. Then quickly cut the part that we injected, and then using the razor blade carefully remove the outer skin so we can see the results. Okay. Alright, so now that I've shown you the procedure, I have a slideshow of pictures that I've taken, and these pictures are high resolution, so it should be easy to determine whether or not water was pushed or pulled. So I'm going to show you the evidence and you're going to decide. Thanks for watching.